The pentose phosphate pathway. In the biochemical pathway, in most animal tissues, glucose is generally catabolized via the glycolytic pathway into two molecules of pyruvate, which is then oxidized via the citric acid cycle to generate ATP. In another metabolic fate of glucose, it generally generates NADPH and specialized products needed by the cell. NADPH is the universal reductant in anabolic pathways. This pathway is called the pentose phosphate pathway, PPP. It is also called the hexose monophosphate shunt. Still others call it the phosphogluconate pathway. In mammals, the pentose phosphate pathway occurs exclusively in the cytoplasm. It is found to be most active in the liver, mammary gland and adrenal cortex. The pentose phosphate pathway carries out oxidation and decarboxylation of glucose 6-phosphate, producing the 5-carbon sugar ribulose 5-phosphate. This pathway is a metabolic pathway parallel to glycolysis that generates NADPH and pentoses as well as ribose 5-phosphate, which is a precursor for the synthesis of nucleotides. While it does involve oxidation of glucose, its primary role is anabolic rather than catabolic. In the first step, glucose 6-phosphate is oxidized into ribulose 5-phosphate and carbon dioxide. During the oxidation of glucose 6-phosphate, NADP plus is reduced into NADPH. The second step of the pathway converts the ribulose 5-phosphate into other pentose 5-phosphates, including ribose 5-phosphate used to synthesize nucleic acids. The third step includes a series of reactions that convert 3 of the pentose 5-phosphates into 2 molecules of hexoses and 1 triose. In the fourth step, some of these sugars are converted into glucose 6-phosphate, so the cycle can be repeated. There are 2 distinct phases in the pathway, the oxidative phase and the non-oxidative phase. In the oxidative phase, NADPH is generated, and in the non-oxidative phase, synthesis of 6-carbon sugars take place. Oxidative phase of pentose phosphate pathway. Here, glucose 6-phosphate is converted to ribulose 5-phosphate and 2 molecules of NADP plus are reduced to NADPH. Step 1. Glucose 6-phosphate is converted to 6-phosphogluconolactone and NADPH plus protonion in presence of glucose 6-phosphate dehydrogenase and NADP plus. There is oxidation reaction of C1 position and hydride transfer to the NADP plus, forming a lactone, which is an intramolecular ester. It is very favorable reaction in the forward direction for producing NADPH. Step 2. 6-phosphogluconolactone is converted to 6-phosphogluconate in presence of gluconolactonase and water and liberates protonion. The lactone is opened by hydrolysis, then 6-phosphogluconolactone is opened up to form gluconate. Often, these types of hydrolysis reactions are very favorable. 6-phosphogluconate reacts with NADP plus and generates D-ribulose 5-phosphate, NADPH, proton ion and liberates carbon dioxide. Step 3. 6-phosphogluconate in presence of NADP plus and 6-phosphogluconate dehydrogenase forms ribulose 5-phosphate through series of intermediates and release NADPH plus proton ion and carbon dioxide. 6-phosphogluconate dehydrogenase performs oxidative decarboxylation, converting the 6-carbon skeleton to a pentose. The C1 carboxylate is removed as the C3 position is oxidized to ketone, making the 5-carbon ketose called ribulose. 2 of the 3 reactions of this first phase generate NADPH. The overall reaction for this process is glucose 6-phosphate plus 2 molecules of NADP plus water reacts to give ribulose 5-phosphate plus 2 molecules of NADPH plus 2 molecules of proton ion and carbon dioxide. The non-oxidative phase of pentose phosphate pathway. In this second phase, 5 carbon sugars or pentoses are generated, particularly ribose, which is necessary for the synthesis of nucleic acids. The non-oxidative phase of the pentose phosphate pathway is composed of 5 steps, but only 4 types of reactions are involved. Step 1. Some ribulose 5-phosphate is isomerized to ribose 5-phosphate through enediol intermediate by pentose isomerase. Step 2. Rest of the ribulose 5-phosphate is converted to xylulose 5-phosphate by phosphopentose epimerase through enediolate intermediate. Epimerization reaction is the flipping of a stereocenter in the substrate. Step 3. Xylulose 5-phosphate and ribose 5-phosphate in presence of transketolase and cofactor thymine pyrophosphate is converted to glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. Thymine pyrophosphate binds at the active sites of enzymes. The amino group of the aminopyrimidine moiety is close to the dissociable proton and serves as a proton acceptor. This proton is promoted by a glutamate residue adjacent to the pyrimidine ring. Thymine pyrophosphate is used to stabilize a 2-carbon carb anion intermediate. Step 4. Glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate further is converted to fructose 6-phosphate and erythrose 4-phosphate by trans-aldolase. Trans-aldolase catalyzes transfer of a 3-carbon dihydroxyacetone moiety from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate. The amino group of an active site lysine residue reacts with the carbonyl C of pseudoheptulose 7-phosphate to form a protonated skiff base intermediate. Aldol cleavage results in release of erythrose 4-phosphate. The skiff base stabilizes the carb anion on C3 and yield the 6-carbon fructose 6-phosphate. This enzyme forms a protonated skiff base intermediate with a ketose, stabilizing a 3-carb anion intermediate, allowing an aldehyde base sugar to react with the enzyme. Step 5. Erythrose 4-phosphate and xylulose 5-phosphate is catalyzed further by transketolase to generate fructose 6-phosphate and glyceraldehyde 3-phosphate. The overall reaction for this process is non-oxidative phase, 2-xylulose 5-phosphate plus ribose 5-phosphate reacts to give 2-fructose 6-phosphate plus glyceraldehyde 3-phosphate. Depending on relative needs of a cell for ribose 5-phosphate, NADPH and ATP, the pentose phosphate pathway can operate in various modes to maximize different products. There are three major scenarios. 1. Ribulose 5-phosphate may be converted to ribose 5-phosphate, a substrate for synthesis of nucleotides and nucleic acids. The pathway also produces some NADPH. Second, glyceraldehyde 3-phosphate and fructose 6-phosphate formed from 5-carbon sugar phosphates may be converted to glucose 6-phosphate for re-entry into the linear portion of the pentose phosphate pathway, maximizing formation of NADPH. Number 3. Glyceraldehyde 3-phosphate and fructose 6-phosphate formed from the 5-carbon sugar phosphates may enter glycolysis for synthesis of ATP. The pathway also produces some NADPH. Ribose 1-phosphate generated during catabolism of nucleosides also enters the glycolytic pathway in this way, after first being converted to ribose 5-phosphate. The sum of these two phases, 3-glucose 6-phosphate plus 6-NADP plus, gives 2-fructose 6-phosphate plus glyceraldehyde 3-phosphate plus 3 molecules of carbon dioxide, 6 molecules of NADPH and 6 molecules of proton ion. The direction of the pathway depending on the body state, ribulose 5-phosphate can reversibly isomerize to ribose 5-phosphate. Ribulose 5-phosphate can alternatively undergo a series of isomerizations as well as trans-aldolations and transketolations that result in the production of other pentose phosphates including fructose 6-phosphate, erythrose 4-phosphate and glyceraldehyde 3-phosphate. These compounds are used in a variety of different biological processes, including production of nucleotides and nucleic acids as well as synthesis of aromatic amino acids like erythrose 4-phosphate. Number 1. Glucose 6-phosphate dehydrogenase. It catalyzes oxidation of aldehyde at C1 of glucose 6-phosphate to a carboxylic acid in ester linkage. NADP plus serves as electron acceptor. Number 2. 6-phosphogluconolactonase. It catalyzes hydrolysis of the ester linkage resulting in ring opening. The product is 6-phosphogluconate. Although ring opening occurs in the absence of a catalyst, 6-phosphogluconolactonase speeds up the reaction, decreasing the lifetime of the highly reactive and thus potentially toxic 6-phosphogluconolactone. Number 3. 6-phosphogluconate dehydrogenase catalyzes oxidative decarboxylation of 6-phosphogluconate to yield the 5-carbon ketose ribulose 5-phosphate. The hydroxyl at C3 is oxidized to a ketone. This promotes loss of the carboxyl at C1 as carbon dioxide. NADP plus again serves as oxidant, that is electron acceptor. Reduction of NADP plus involves transfer of 2 electron plus 1 proton to the nicotinamide moiety. This difference has little to do with redox activity but is recognized by substrate binding sites of enzymes. It is a mechanism for separation of catabolic and synthetic pathways. Number 4. Phosphopentose isomerase converts the ketose ribulose 5-phosphate to the aldose ribose 5-phosphate. Number 5. Phosphopentose epimerase interconverts the stereoisomers ribulose 5-phosphate and xylulose 5-phosphate. Both reactions of epimerase and isomerase involve deprotonation to form an N-diolate intermediate, followed by specific reprotonation to yield the product. Both reactions are reversible. Number 6. Transketolase actually transfers an aldol moiety, that is glycoaldehyde, a 2-carbon fragment from xylulose 5-phosphate to either ribose 5-phosphate or erythrose 4-phosphate. Transketolase utilizes a prosthetic group thiamine pyrophosphate, a derivative of vitamin B1. The positively charged N in the thiazole ring acts as an electron sink, promoting carbon-carbon bond cleavage. The 3C-aldose glyceraldehyde 3-phosphate is released and 2C fragment remains on TPP. Number 7. Trans-aldolase catalyze transfer of a 3-carbon molecular fragments respectively in each case from a ketose donor to an aldose acceptor. Completion of the reaction is by reversal of these steps. The 2-carbon fragment condenses with one of the aldoses erythrose 4-phosphate 4-carbon or ribose 5-phosphate 5-carbon to form a 6-carbon or 7-carbon ketose phosphate product. Transfer of the 2-carbon fragment to the 5-carbon aldose ribose 5-phosphate yields pseudohaptulose 7-phosphate, while transfer instead to the 4-carbon aldose erythrose 4-phosphate yields fructose 6-phosphate. Thus, the pentose phosphate pathway serves as an entry into glycolysis for both 5-carbon and 6-carbon sugars. Glucose 6-phosphate dehydrogenase is the first irreversible committed rate limiting step of the pentose phosphate pathway. It is allosterically stimulated or regulated by NADP+ and strongly inhibited by NADPH. So, when the cytol concentration of NADPH is high, the enzyme's activity is low. It is also allosterically regulated by fatty acid acyl esters of coenzyme A. An NADPH utilizing pathway forms NADP+, which stimulates glucose 6-phosphate dehydrogenase to produce more NADPH. This step is also inhibited by acetyl coenzyme A and is stimulated by insulin. Glutathione peroxidase catalyzes degradation of organic hydroperoxides by reduction as 2 glutathione molecules are oxidized to a disulfide. Regeneration of reduced glutathione requires NADPH produced within erythrocytes. This process can also control the pathway. Glutathione reductase also requires NADPH. The ratio of NADPH is to NADP+ is normally about 100:1 in liver cytosol, making the cytosol a highly reducing environment. Thus, it can also regulate the pathway. The pentose phosphate pathway is one of the 3 main ways the body creates NADPH, approximately 60% in humans. While the pentose phosphate pathway is involved, oxidation of glucose, its primary role, is anabolic and synthesizes large complex molecules from small precursors. Additionally, NADPH can be used by cells to prevent oxidative stress. NADPH reduces glutathione via glutathione reductase, which converts reactive H2O2 into water by glutathione peroxidase. RBCs and tissues that carry out extensive fatty acid synthesis like liver, adipose tissue, lactating mammary gland, or actively synthesizes cholesterol and steroid hormones as in liver, adrenal glands and gonads require NADPH provided by this pathway. Rapidly dividing cells such as those of bone marrow, skin, intestinal mucosa and tumors use pentose ribulose 5-phosphate to make RNA, DNA and coenzymes as ATP, NADH, FADH2 and coenzyme A. This can also be used for the catabolism of pentose sugars from the diet, the synthesis of pentose sugars for nucleotide biosynthesis and the catabolism and synthesis of less common 4 and 7 sugars. In addition to ATP, the dark reactions of photosynthesis require reducing power in the form of NADPH, which is the currency of readily available reducing power in cells. The pentose phosphate pathway and the Calvin cycle have in common several enzymes and intermediates that attest to an evolutionary kinship. The Calvin cycle uses NADPH to reduce carbon dioxide to generate hexoses, whereas the pentose phosphate pathway breaks down glucose into carbon dioxide to generate NADPH. The Calvin cycle is sometimes referred to as the reductive pentose phosphate pathway. Disorders related to the pathway: genetic deficiency of glucose 6-phosphate dehydrogenase can lead to hemolytic anemia because of an inadequate supply of NADPH within red blood cells. It affects around 400 million people around the world. Wernicke-Korsakoff syndrome is a disorder caused by as severe deficiency of thiamine. It is common in alcoholic people. This syndrome is caused by a genetic defect in transketolase, which lowers the affinity for thiamine pyrophosphate. The symptoms include severe memory loss, mental confusion and partial paralysis. The main purpose of the pentose phosphate pathway is to regenerate NADPH from NADP+ through an oxidation reduction reaction. This reaction is coupled to the formation of ribose 5-phosphate from glucose 6-phosphate. NADPH is used for reductive reactions in anabolism, especially in fatty acid synthesis. The non-oxidative portion of the pathway creates carbon chain molecules ranging from 3 to 7 carbons. These compounds are intermediates in glycolysis and gluconeogenesis or other biosynthetic processes. The pentose phosphate pathway also produces fructose 6-phosphate and glyceraldehyde 3-phosphate.